talked about the loop, and it talked about this concentration, the ability for the loop to concentrate the tubular fluid, and this is where we're headed today. So that was really, that video was combining the effects of the cortical nephrons and the juxtamedullary nephrons. And we're going to distinguish between the two. So we get a little bit more in depth in that video. But it was a great start for appreciating how the kidney works. And so we did talk about all the parts of the kidney and the parts of the functional unit of the kidney called the nephron. And we're going to get in more detail about how that nephron works. And next week, I think it's Wednesday, we're going to have a little group exercise where you will be asked in your group to draw a nephron and describe what happens to specific molecules, sodium chloride, potassium, water, glucose, etc., within the nephron regarding filtration, reabsorption, secretion, and water conservation. So pay attention now and start thinking about how you're going to put your group presentation together. You're going to be doing it on the board. You're going to be drawing out the nephron, however creative way you want to display what the nephron does. Okay, so the nephron is responsible for filtering the blood, as we've already talked about. Remember my analogy that you're moving and you have to pack everything up and you decide which things you're going to save or which things you're going to get rid of. And there are certain things that you can't move. And those big things include the washer, the dishwasher, the refrigerator, whatever big appliances you have in your space that you can't take with you. What do those represent in terms of the blood? Does anybody remember that? No?
Proteins, right?
Albumin?
Albumins, not so many, because they're kind of small.
Oh, really?
They're smaller proteins.
Oh.
Some of them can get through, but you're on the right track.
Red blood cells, white blood cells?
Red blood cells, white blood cells, larger
Larger
proteins, and
Plasma.
it begins with a P.
Yes.
Platelets.
That's it.
Yes.
Like, no,
So the glomerulus, the cluster of capillaries within the renal corpuscle, do the filtration. These capillaries are fenestrated capillaries, so they've got little holes penetrating all of the endothelial cells. And materials can get pushed out of those cells, out of those holes, from the blood into the space called the glomerular capsule. And that space collects what's known as the filtrate. And that filtrate contains virtually everything that can be pushed out of the blood that can be, except for the red cells, the white cells, the large platelets, the large proteins, and the platelets. So glucose, sodium, chloride, potassium, urea, everything gets pushed out into the filtrate, except for those structures that we just talked about. And that fluid is very similar in its osmotic activity, osmotic pressure, and its millimoles equivalent, except of course it is completely different to the blood. The blood has all these other things in it, whereas filtrate doesn't. And the filtrate has things that the blood doesn't. And so that filtration process produces a fluid, and then that fluid travels through the tubules. Remember, we have a proximal convoluted tubule that was really long and tortuous, twisted, right? And in the proximal convoluted tubule, we see a lot of reabsorption. The reabsorption is those things that you put in the boxes that you thought you wanted to get rid of. Oh my God, no. You need to retrieve them, right? All those records that you thought you would, no, keep those. Keep your records. I'm not talking LPs, I'm talking taxes and driving stuff and automobile records and all those things you want to keep. So you got to go bring those back in. And that's what happens in tubular reabsorption. Those substances that we want to retrieve from the filtrate are pulled back into the blood along the proximal convoluted tubule. Then we have down here, and particularly between the proximal convoluted tubule, we have this really important section of the nephron called the loop. It's also known as the loop of Henle. So somebody named Henle first identified it. The nephron loop, or the loop of Henle, has some specialized activity going on. There's a thick descending and a thin descending part, then there is a thin ascending and a thick ascending part. And in the thin descending part, we can see reabsorption of water. Water, it's permeable to water, but not much else. And then as we get up and head toward the ascending part, that portion of the loop is not permeable to water, but it is permeable to sodium and chloride of potassium. And wait till you see what happens if your potassium levels are out of whack outside the cell, big trouble. And so the kidneys are really important for managing that extracellular potassium level within the blood. That loop is really important for water conservation. And the vasorecta, which is the long one that's part of the juxtamedullary nephron, that part of that loop is all about water conservation. Because wherever you have sodium and chloride being pulled back from the tubular fluid into the interstitial space and then back into the blood, you're going to have water following it. And so that water coming out of the bottom of the loop is actually coming out of the tubular fluid in response to the increase in sodium and chloride that we put back in the blood. It'll become clearer as we go along. As we get up toward the distal convoluted tubule, we have less reabsorption and more secretion. What secretion is things that didn't necessarily get filtered out of the blood initially are filtered out of the blood here at the distal convoluted tubule region. Water conservation is a big deal here. This is what our kidneys are designed to do. Filter the blood, get rid of the waste, and then save the water. Save the water. Thank goodness, because did I remember my water today?
Mm.
No. Okay. So we are going to pay attention to how all of this goes on. In the glomerulus, we have this collection of capillaries. The capillaries are fenestrated, and these capillaries are surrounded by some specialized cells called podocytes. Can anybody imagine why they would be called podocytes? What is a podiatrist? A foot doctor. What do these look like?
I guess little feet.
Little feet, little toes or little feet.
Like
Yes. So
tentacles.
this podocyte has little extensions, and these extensions, they interdigitate with each other. And they form a slit called a filtration slit. So here we're looking at the endothelium and the fenestrated holes of the endothelium. We have a basement membrane here, basically a protein, proteoglycan, a little sugar in there. And then we have, these are the feet, the little pedicels of the podocytes. We have bigger holes of the fenestrated capillaries than we have filtration slits. And you can see here that certain things do not make it through the fenestrations, all these large things that we talked about, especially if you have something that's hooked onto a protein, like some of the hormones. Remember, you have to have a protein transporter because they're lipid soluble and they have to be carried in the blood by a protein. And so those things they don't get through, they stay in the blood. That which gets through has to be able to get through the slits too. And these are all the sorts of things that can make it through the slits. The electrolytes, sodium, chloride, potassium, magnesium, calcium, et cetera, et cetera, et cetera. Water, big deal. And water, by the way, isn't moving on its own. What is it that draws water? Solutes. Any of these can be osmotically active, including and especially considering glucose. So if glucose is in the blood, either you've had you've just eaten, or you're not utilizing that glucose in some way, and that would indicate that you have perhaps diabetes. If your blood is carrying too much glucose, that glucose is going to end up in the filtrate. And if you don't need that glucose, and you have so much of it in the blood because you have diabetes and you're not able to take it out of the blood, it's going to end up in the urine. And it's going to draw water with it. And people who have diabetes, mellitus, have a condition called polydipsia polyuria.
You have to pee a lot.
They have to pee a lot. Why do they have to pee a lot? Because they have an osmotically active substance in the filtrate that's drawing water, keeping water in the filtrate. And if you aren't conserving your water, what does that do to you?
Dehydrated.
You get dehydrated, and your hypothalamus says, go drink, because you're dehydrated. So, we see these things that are ending up in the filtrate. many of these things get pulled back in. The water typically gets pulled back in. We don't produce nearly as much urine as we have water filtering through the kidneys at any given time. Our amino acids can get pulled back in, fatty acids, vitamins. Remember those A, D, E, and K, the ones that you gotta have fats to absorb? Well, then we have the water solubles. And if you take too much vitamin C, what are you gonna do? You're just gonna pee it out. So think about that next time you're spending money on your vitamins. Urea, uric acid, and creatinine, what are these considered?
Nitrogenous waste.
Nitrogenous waste products, yes. So we're gonna see how the podocytes and the capillaries together form this filtration membrane. Filtration membrane, keeping blood in, keeping blood cells in, keeping the proteins in, but then moving everything else out so that then they can be selectively reabsorbed back into the blood. These are scanning electron micrographs of your podocytes. Don't you love that beautiful imagine of the way they are interdigitating with each other? And here's fenestrated capillaries, so it gives you a nice idea of how those holes are dotting, just covering the walls of those endothelial cells so that we can get that stuff out. This is just a diagram of one very important structure that I want you to pay attention to. And this is this distal tubule, this distal convoluted tubule, and the collection of chemoreceptor cells, collectively known as the macula densa. And they call it dense because they're packed in there really densely. And this is where we can determine whether or not we have low blood pressure or high blood pressure based on sodium and chloride concentrations. If we have high sodium chloride concentrations, we probably have high blood pressure because there's more blood flowing through and carrying more of that sodium chloride along with it. And if we have low blood pressure, maybe we have low sodium and chloride content, and the kidneys will act accordingly. Here's our capsule. The capsule is made of squamous cells, and this is considered the parietal layer of this whole structure. The podocytes are considered the visceral layer of this renal corpuscle. So the renal corpuscle has a parietal layer and a visceral layer via those podocytes. It's also showing you some specialized cells within the glomerulus called mesangial cells. We're going to learn more about those mesangial cells and how they can help to control blood pressure within the capillaries. We have an afferent arteriole coming in, we have an efferent arteriole leaving, and remember that our afferent arteriole has a larger diameter, feeding blood into this glomerulus than the efferent arteriole does, or efferent arteriole does. That fluid that is created by the filtration process is then going to leave the capsule and head out through the proximal convoluted tubule. So look at the amount of blood that's going through our kidneys on any given day. That's an enormous amount of blood in a day. Of course, these are approximates. Everybody's going to have a little bit of difference. Number of liters that the filtrate is produced by from that. Still, can you imagine having to pee 180 liters every day? That'd be crazy. You'd never leave the bathroom. You'd be drinking one and out the other. We have, interestingly enough, the same milliosmoles, the same pressure on both sides, blood versus filtrate, except that they are different in their composition, as you can imagine, right? When we have this filtrate, we're taking everything that we can pull out of it via those little holes, those little fenestrations, and then the large things have to stay back. So those large proteins stay back. Some of the small albumins can cross, but not all of them, smaller proteins. And anything like you can see here that's bound to a protein is probably not going to get through the filtration process. So filtration is a really important product because it has to do with hydrostatic pressure within the blood itself. And this is where we're heading. So we have here our efferent arteriole bringing blood in. We have efferent arteriole blood leaving. Inside here we have these capillaries, the fenestration, and you can imagine that if we have a larger flow of blood coming into the capillaries and a narrower flow leaving, we're going to build pressure in the glomerulus itself. And that pressure in and of itself is going to push those molecules, those electrolytes, those products, those particles out of the blood. It's called blood hydrostatic pressure. It's the greatest force that is pushing the material out of the blood. We have a colloidal osmotic pressure that's opposing it, and that's why they say it's negative. This colloidal osmotic pressure is going to draw water, back into the blood in the capillaries. Because proteins are osmotically active. So they're going to pull that water back into the glomerulus a little bit. So we're going to take our blood hydrostatic pressure and we're going to subtract 32 from the blood hydrostatic pressure. We're also got this the capsular pressure, which is pretty negligible. It's just 18 out of the total 18 millimeters of mercury. But this capsular pressure can have minor impact. And so we're going to, it negatively affects, I think the most important part is to remember that the capsular pressure and the colloidal osmotic pressure negatively impact the blood hydrostatic pressure. And this little formula right here is telling us that if we take the negatives away from the positive pressure, we're going to end up with 10 millimeters of mercury. This is what's known as your net filtration pressure. Why do we care about this? Well, again, it's all about pushing the material out of the blood so that we can put it into the filtrate, into that tubular fluid, and then reabsorb all the important things via the tubes, the proximal convoluted, the loop, and the distal convoluted tube. So our hydrostatic pressure is what's the driving force of pushing everything out. These negate it a little bit, and ultimately we're going to remove these from this total and get our net filtration pressure. And our net filtration pressure is going to determine our glomerular filtration rate. Or rather, our glomerular filtration rate is going to affect our net filtration pressure. So what's this say? Yes. That's just talking about what I just talked about, right? Mm-hmm. Yep, so we want to know how we can manipulate these numbers. So remember that your blood hydrostatic pressure is going to be your strongest outward force. These two are going to negate a portion of this blood hydrostatic pressure because they're pulling water back in. And so we're going to have a net filtration pressure as a result of subtracting these from that. All right, so now we're going to talk about the glomerular filtration rate. And the glomerular filtration rate is impacted by your net filtration pressure. Okay? As we can see, we have an average of 125 mils per minute, meaning this is what's produced every minute by your kidneys, filter-wise, fluid-wise. Quite a bit, isn't it? 125 mil? It's a good amount of fluid in a minute. Some of this is converted to urine, but the rest is reabsorbed. So we can't possibly have to be able to pee 125 mils that is produced every single minute. That would just be crazy. So we're going to see this ability for our kidneys to bring more water back into the system and just get rid of the bare minimum. The bare minimum. All right, so one of the things that the physicians are going to look at in terms of how your kidneys are working is your plasma creatinine. We did talk about blood urea nitrogen. That's another measurement that they use. But your plasma creatinine is also going to be an important factor for determining how your kidney is functioning. So the blood plasma creatinine is typically very consistent. It's very constant. You take creatine phosphate, you make the creatine phosphate, and then when you need that extra phosphate, you cleave the phosphate off the creatine and you attach it to ADP. And the leftover is creatinine. And if you have too much of this creatinine in the plasma, it's going to spill over into the urine. And you can see here that if you have a greater than 135 millimoles of this creatinine in the blood, you're going to see that creatinine in the urine. Greater amounts of it in the urine. Your glomerular filtration rate, as I said earlier, is going to be impacted by your net filtration pressure. And as this says, anything that affects your net filtration pressure is going to impact your glomerular filtration rate. What's the glomerular filtration rate? The amount of fluid that is coursing through your kidneys, pulled out of the blood in every minute. in any minute. All right, so if we have a decrease in our renal blood pressure, that's decrease in blood pressure overall, decrease in our renal blood pressure, then we're going to see a decrease in the net filtration pressure. Why? Because your blood is coming in at a certain pressure. If it's lower, then we're going to have a lower blood hydrostatic pressure. And if we have a lower blood hydrostatic pressure, we're still going to have these guys, but we're also going to impact and lower the net filtration pressure. Everybody see that? Good, good, good. Things can happen. You can have reasons for that lower blood pressure. Maybe you have a hemorrhage going on. Maybe you are dehydrated. All those things would impact your blood pressure overall. Maybe you have a decrease in the ability of your kidneys to filter, and if so, you can lead to a condition called azotemia. Do you remember what we talked about with azotemia? Azotemia is a laboratory finding, but it's a result of what? Do you remember, Yulia?
Was it too much nitrogenous waste?
The urea. Increased urea is going to give you azotemia. Why? Because urea is a waste product. And urea, remember, is in front of urea is ammonia. And so if you have a urea that is not being filtered out effectively by the kidneys, it's going to back up into the blood. You're going to have this, like the sewage, your septic system. Anybody have a toilet that's overflowed?
No.
I think of that every time I think of urea getting backed up into the blood. So, your glomerular filtration rate is going to be determined by your net filtration pressure. And your net filtration pressure can be impacted by your blood pressure. So if you decrease your blood pressure, you're going to decrease this and you're going to decrease this. And if you have a lower glomerular filtration rate, that means that things don't get filtered out as well. If you have an increase in your glomerular filtration rate, why would your glomerular filtration rate increase? Maybe you have an increase in blood pressure, increasing the rate at which things are pushed out of the blood, increasing the production of that filtrate. And that is going to increase your net filtration pressure. And that is going to lead to perhaps fluid flowing through your kidneys too fast, because you've got this big flush of fluid coming out of the glomerulus and into that proximal convoluted tubule. And then that fluid is rushing through the tubules and the loop and back out again into the collecting ducts. And that means that you might not reabsorb all the things that you need to reabsorb. So, as it says, maybe you have a depletion of your sodium and your chloride. Maybe you have potassium that stays in the blood and you don't get that secreted out. Reabsorbed, I should say. Secreted out. Maybe you have dehydration. Yeah, because you're losing too much water. So the kidneys have some mechanisms to counter these potential situations. Several mechanisms the kidneys use to manage the blood pressure coming into the glomerulus and thus leaving the glomerulus and producing that fluid. We know that we have both auto or intrinsic regulation and extrinsic regulation happening in the kidneys. And the first of the two autoregulatory or intrinsic mechanisms is referred to as myogenic mechanism. And this is so simple. Remember that you have blood coming in into the glomerulus and then back out again. And when we have this myogenic, we know that we have little smooth muscle cells surrounding our arterioles here, right? They're not just endothelial cells. There's a little bit of smooth muscle around arterioles. And what happens when you stretch smooth muscle? It wants to constrict. So very simply, if you increase the flow, there's our increase in our blood pressure, then we're going to have a myogenic effect right here at the site. These little smooth muscle cells are going to constrict, and they're going to reduce the diameter of our afferent arteriole, and that means it's going to reduce the flow of blood into the glomerulus. Very clever. By the same token, if you have a decrease in your blood pressure, then those smooth muscle cells have no reason to constrict. They're going to relax, and that's going to increase the diameter of the afferent arteriole, which means that more blood can flow in to the glomerulus, hence affecting our glomerular filtration rate. Ultimately, this is what we're talking about, right? Okay. The next mechanism is called the tubuloglomerular feedback autonomic autoregulatory system. So imagine this, as this diagram says, we have a high glomerular filtration rate. Why would we have high glomerular filtration rate? Increase in blood flow, right? So let's say that the myogenic isn't enough to counter the high blood pressure. We have this increased flow of blood into the glomerulus. That's going to increase the filtrate process, because if we have an increase in the flow of blood in, it's going to increase the blood hydrostatic pressure, it's going to increase the net filtration pressure, hence the glomerular filtration rate is going to increase. Can everybody see that? Good. So we have an increased flow of blood, which is going to increase the filtrate, and then we have this little collection of cells called the macula densa, and the macula densa cells say, oh my goodness, there's a lot of sodium and chloride in this filtrate that's coming through the tubules. Because remember, it was all pulled out and put into the fluid. And that is going to cause something called a paracrine secretion. What's a paracrine secretion? This is a secretion of a hormone right there at the site. And the hormone right there at the site is, believe it or not, ATP, which is neurotransmitter, but in this case, it's considered a hormone. This ATP is going to ultimately result in a constriction of the afferent arteriole. It's going to slow the blood flow, it's going to slow the glomerular filtration rate, it's going to reduce the net filtration pressure back to normal. So our flow, the key is we want the tubular fluid to flow at an even rate, regardless of the blood pressure that's feeding those glomeruli. We want a steady flow so that we can reabsorb the materials that we want and need and get rid of the waste. If it flows too slow, it's stodgy. If the flow is too fast, we lose those electrolytes that we want to recoup. All right, so we got the the myogenic, which is taking care of right there at the site of the blood afferent arteriole, and then we have the tubular glomerular feedback mechanism. And this right here is where those macula densa cells are located. This is a section of the distal convoluted tubule. It's tucked into this, what they call the vascular pole for the glomerulus. And these cells, little chemoreceptors that tell levels of sodium and chloride. They absorb them and then they report to this process called the juxtaglomerular apparatus, the JGA. There are 3 different components for the JGA, and that is that we have some specialized smooth muscle cells called the juxtaglomerular cells. You can also call them granular cells or G cells. And we have the macula densa right here in the distal convoluted tubule. And we also have these little specialized cells called the mesangial cells. And I mentioned those earlier. They can manage our capillary pressure constriction, and then they can manage this, what's happening right here. So, the macula densa in the distal convoluted tubule is capable of detecting sodium and chloride content in the tubular fluid. And by doing that, they can tell these cells and these cells what to do. And so the macula densa is going to release ATP, and it's going to, that ATP is going to be converted into adenosine, by the mesangial cells, and the adenosine has a special effect. I want to also remember to report to you that the mesangial cells, or the macula densa, is capable of producing erythropoietin. And what's erythropoietin all about?
Trigger red blood cells.
Trigger red blood cell production. So doesn't that make sense? If you have low blood pressure, these cells detect low sodium and chloride content as well as high sodium chloride content. If we have low blood pressure, what do we want to do? Make more red cells. Because that's part of how you raise the pressure in blood. Make more red cells. Okay, so the mesangial cells are then going to respond to the ATP that was released by the macula densa, right? ATP released. The mesangial cells convert that ATP to adenosine and The adenosine tells these granular cells to contract and constrict the afferent arteriole, which will raise blood pressure, or not. These G cells also release renin, and renin is responsible for correcting low blood pressure, in part. We have lots of ways of doing it. So the adenosine, from the mesangial cell conversion of ATP to adenosine, this adenosine is going to trigger these G cells or the juxtaglomerular cells to constrict and then reduce the flow if there's high blood pressure. If there's low blood pressure, then these G cells are going to release renin, and the renin is then going to have a widespread effect over all the body. What about the nervous system? Here's what I remember about the nervous system and the kidneys. I don't want to have to pee while I'm in conflict, right? Now, maybe you have a really full bladder, and suddenly, like a dog that's really nervous will pee, a horse, all these, we all do it. You have a really full bladder, you're going to get rid of that, but you're not making any more urine, is the point. Because the sympathetic system slows all that stuff down. Why? Because blood is going to be diverted to where? Where do you need your extra blood when you're in conflict? Skeletal muscles, right. The core, skeletal muscles. Got to be able to get away from things. And so your blood pressure is going to drop in the kidneys from the sympathetic innervation. The goal is to decrease the glomerular filtration rate, thus decrease the output of urine, and that makes total sense. Total sense. Now, there is a release of norepinephrine. Maybe you remember norepinephrine has some wide, wacky effects. And you can see here this norepinephrine also is going to affect your afferent arterioles by contracting the granular cells, slowing down blood flow into the renal tubules, or the glomeruli. And there we have it. Another sympathetic stimulation of the granular cells causes them to release renin. So we've got plenty of influence on those from the kidneys to go to the renin, angiotensin aldosterone system. And the renin, angiotensin aldosterone system, as you can see, starts right here in the kidneys by the production of renin. Renin then is carried through the blood, and it converts a prozyme, angiotensinogen, in the liver, converting that angiotensinogen into angiotensin 1. Angiotensin 1 travels through the blood and hits the capillaries of the lungs, where there's an enzyme called angiotensin converting enzyme. Maybe you've heard of this as a really common blood pressure medication. Very common for hypertension, called an ACE inhibitor. Now you can see why it works. If you inhibit the ACE, you don't change angiotensin 1 to angiotensin 2, and you don't get vasoconstriction, which then reduces your blood pressure. Because the more you constrict blood vessels, the higher the pressure on the blood. Back up to the heart. So you got to remember the heart's in all of this. Heart's pumping all that blood around. So when we have a normal system going on, and we don't have high blood pressure, and we're not using an ACE inhibitor, this angiotensin converting enzyme converts angiotensin 1, angiotensin 2, and then angiotensin 2 does all of this stuff. First, widespread vasoconstriction. So all the arteries, arterioles constricted. And that's going to increase blood pressure. Remember, renin is released in response to low blood pressure. And we have angiotensin 2 impacts the adrenal cortex. What happens in the adrenal cortex? Let's see, we have 3 zones in the cortex of the adrenal gland. Who remembers what those zones are? From the capsule to the medulla. What's the name of the first one out there by the capsule? Zona glomerulosa, yay! What does zona glomerulosa produce that we are primarily interested in? What hormone?
Is it aldosterone?
Yes, aldosterone. Mm-hmm. And by the way, I think I said that aldosterone was protein-based, but no, it's not. It's one of the 11 steroid. So it's one of the lipid-based hormones. So scratch that from the notes, will you, Nikki? So aldosterone is produced in the adrenal cortex via the zona glomerulosa. Aldosterone hits the kidneys, and in the ascending limb, aldosterone actively then influences the ascending limb cells to actively transport sodium and then chloride and potassium from the tubular fluid back into the blood. Then, on the descending limb, you got more sodium and chloride in the interstitial spaces, and then ultimately heading to the blood, pulls water out of the descending limb, that thin descending limb, right? Because water goes wherever there's higher sodium and chloride concentration, especially. So we got that. What's that going to do to our blood? Raise the blood pressure, because we add water to the blood. Then we also have an influence on the hypothalamus, and where aldosterone, or excuse me, this angiotensin 2, this hits the hypothalamus. The hypothalamus says, oh my gosh, that blood pressure must be really low. You need to go drink some water, Heather.
You should leave a bottle here for yourself.
It would never last, right? So, very interesting how that all works. So you got to remember your renin, angiotensin, aldosterone mechanism for responding to low blood pressure. And if your blood pressure is low, your glomerular filtration rate is low, and that's going to slow the fluid coursing through the kidneys, makes everything really stodgy. Aldosterone. How does the hypothalamus respond to that thirst? Oh, let's review that. What is it that in the hypothalamus, what does the hypothalamus produce that impacts our thirst mechanism? It's posterior pituitary, I'll tell you that. So the hypothalamus and the posterior pituitary are in cahoots, so to speak. What is it that the hypothalamus produces that's stored in the posterior pituitary? ADH. What's ADH stand for?
Antidiuretic hormone.
Antidiuretic hormone. And what does that mean?
No peeing hormone.
No peeing, right?
Yeah.
Because when you have ADH, then we have specialized cells in the collecting ducts and the distal convoluted tubule called principal cells. And those principal cells have little proteins embedded inside them called aquaporons. Some of those aquaporons are permanent, so we're always pulling water through those aquaporons. Remember, water is a large, we can readily diffuse it, but it takes a little longer, so it's best to have pores, aquaporons, that can pull that water through. And then those principal cells respond to ADH by creating more aquaporons in the distal convoluted tubule and the collecting ducts, and we retrieve even more water that way. So we don't pee as much. Antidiuretic hormone. Something else that happens in our kidneys is this hormonal response. The hormonal response is the renin and the atrial natriuretic peptide and the brain natriuretic peptide. And so what we've been talking about so far is pretty much low blood pressure, but what if we have really high blood pressure? How does all of that work? So you might remember from 232, did we talk about this? We did in the endocrine system. We talked about atrial natriuretic peptide. It is produced in the atria of the heart. When the heart muscle is stretched farther than it should be because there is hypertension, greater blood flow, then this heart produces this atrial natriuretic peptide in response to that increased blood flow, and that ANP then causes the afferent arterioles to relax. And if we increase the diameter of the afferent arteriole, we increase the blood flow. If we increase the blood flow, we increase the glomerular filtration rate. If we increase the glomerular filtration rate, we're going to not recoup as much water because it's flowing really fast, along with the ADH, right? Yeah, and getting rid of extra water. Relaxes the mesangial cells. What do the mesangial cells do? Mesangial cells, let's see, they convert ATP to adenosine, and what does adenosine do? Adenosine triggers the granular cells to constrict, but we don't want that because we want that blood to flow, and so we're going to inhibit the granular cells, inhibit the release of renin. Because renin is response to low blood pressure, and this is response to high blood pressure. So let's see. So we have relaxing mesangial cells, increasing surface area filtration memory. What effect on the glomerular filtration rate, what is this going to have on the glomerular filtration fluid volumes? If we relax the afferent arteriole, constrict the efferent arteriole, what's it going to do? I still can't hear you. Decrease? If we increase the glomerular filtration rate, relax the afferent and constrict the efferent, we're going to have greater push. So we're going to actually see an increase in the fluid production, and therefore an increase in ultimately urination, because that fluid is going to, we're not going to recoup as much water, we don't need it. And the idea is to get rid of the extra water, to reduce the blood pressure, right? Yeah. If we inhibit the release of renin, what effect does this have on the downstream chemical, ultimately the glomerular filtration fluid rate? Well, again, we're not going to have the constriction of those blood vessels overall. We're not going to see that same fluid flow. We're going to see an increase in fluid flow. We're going to see a decrease in blood pressure as a result. So that's tubular filter. That's the filtration aspect. So we have reabsorption that takes effect along the length of the tubules, but the vast majority of the reabsorption occurs with the proximal convoluted tubule. And the proximal convoluted tubule is super long. Remember we looked at the histology? No, we didn't. We're going to do that on, not this Wednesday, next Wednesday. Can you believe it? A whole week away. What we have to keep in mind is that this tubular reabsorption happens along the length of our, I guess I don't have a picture of it right here. Along the length of particularly the proximal convoluted tubule, some in the loop, and very little reabsorption that happens in the distal convoluted tubule. But this tubular reabsorption is an important component because once all that stuff has been pushed out of the blood and into the fluid, we have to recover most of it. So we don't want glucose heading off to the urine if we're going to use that glucose. So we reabsorb glucose. We want our sodium and chloride to be reabsorbed so that we retain our water. We are going to reabsorb potassium in the tubular reabsorption in the proximal convoluted tubules, but then we're going to secrete it in the distal convoluted tubules to keep our potassium levels right in extracellular fluids. This reabsorption in our proximal convoluted tubule in particular, and in the rest, but in particular in the proximal convoluted tubule, can happen a couple of ways. It can happen through the cells, as in transcellular, or it can go around the cells, as in paracellular. And even though these cells that are lining the tubules, by the way, they're cuboidal epithelial cells with a lot of microvilli on them, even though they are held together by tight junctions, stuff gets through. And stuff gets through via something called solvent drag. Solvent drag, where water can actually drag lots of different molecules along with it. So in between the cells, we have paracellular transport. But for the most part, we want things to go through the cell itself, transcellular transport. Lots of proteins, of course, are essential for doing this. We have some facilitated proteins that allow things to just pass right through, but we also have some proteins that are called symports that utilize sodium to pull something else along. And then we also have some proteins that are called antiports, where one thing will be shipped in and another thing will be shipped out. So let's see how that works. First, I want to make note of all of our little microvilli. And so when we look at the tissue in the microscope, when we get to class next Wednesday, a week and a half from now, we're going to look at the histology of the kidney, and we're going to see all these little tubules that look like they're kind of fuzzy on the inside, and then the occasional tubule that's nice and clean. And we can really identify the cuboidal epithelial cells that make it. The nice clean ones that are less frequent are the distals, and the ones that are kind of fuzzy looking on the inside, those are the proximal convoluted tubules. We have many more of them than we do the distal convoluted tubules. So this fuzzy microvilli give the luminal surface of these tubes kind of a fuzzy appearance. So you see here, most of the filtrate is filtered and brought back into the blood via reabsorption in the proximal convoluted tubule. We got some mitochondria that are capable of producing ATP, and they are driving the pumps, those protein pumps. So here's our explanation for all of it. Here's our tubular cells. Here's our blood. Here's the tubular fluid, the interstitial fluid, and here's the tubular fluid itself. Did I say tubular fluid? I meant tissue fluid.
Yes.
I meant tissue fluid. You know, interstitial fluid. So we have our tubular fluid flowing through. We got sodium, we got sodium, we got chloride, we got all these other things, calcium, potassium, magnesium, uric acid, urea, all of these things are coursing, glucose, all these things are coursing through the blood, they get pulled out of the blood, put into the tubular fluid, and then we want to reabsorb them. And so you can see here we have a protein called a symport that's going to be using sodium to move glucose into this tubular cell. We're going to use these things called antiports to move hydrogen out. Where does the hydrogen come from? Here and here. So our kidneys are actually important for helping us maintain correct blood pressure, pH, as well as blood pressure. Sodium in, potassium or hydrogen out. We're going to use another antiport for moving our negatively charged ions out, and our chloride in, sodium and chloride, back to the blood. Why do we want sodium and chloride back into the blood? Because it draws water. Why do we want glucose back in the blood? Well, because we don't want to waste our glucose unless there's way too much of it. And if there's way too much of it, it means there's something going on in your body if you have glucose spilling over into your urine. Now, in these days, they don't even wait to see glucose in the urine. That was like old technology and identifying diabetes that way. Now they'll find excessive levels of glucose in your blood, and they might have you do I don't call it starvation, but fasting blood sample, right? Where you go to instead of eating breakfast, so the last thing you ate was dinner, you go and have your blood drawn first thing in the morning. They want to see what glucose levels are like on a fasting phase. So we're going to glucose in with sodium, we're going to move water in via aquaporins, they're permanent, and then we're going to move these things into the tissue fluids and back into the blood. Glucose gets its own little facilitated transport. Sodium, of course, is going to be pumped out of the cell, and then potassium back in. Why? Because we want our sodium-potassium levels to be that way. And we're going to use the sodium-potassium pump to do that. That's where the mitochondria come into play. You can see that our potassium is still going to be used to move chloride out through the symports. And again, we're just going to keep moving that potassium back in via these sodium-potassium pumps. And we have that solvent drag that helps move things that don't get through the cells, but through the spaces. Glucose, super important to pull the glucose out of the tubular fluid back into the blood. You see those small amino acids can get involved there as well. So lots of things are going to be moved either through the cells or between the cells. Solvent drag. So that's the proximal convoluted tubule. Then we have the loop, the nephron loop. And you might remember the nephron loop had a thick descending portion, and then a thin descending portion, a thin ascending portion, and a thick ascending portion. So my references and all the work of research I've done is, we're not going to worry about that thin ascending part, okay? We know that the thick descending part is going to be still involved with reabsorption of all of our materials that we want to pull back in. Then we have the thin descending part, which is permeable to water. And it's permeable to water because it's made up of simple squamous epithelial cells. But they're special. Look, they even have their own little microvilli. So, not so many solutes get through that thin descending part, which means that water is freely flowing. But what does happen is that we have reabsorption along the thin or thick ascending portion, and that reabsorption at the thick ascending portion is all about sodium, and chloride, and potassium. And when sodium and chloride are reabsorbed from the tubular fluid into the tissue fluid, what are they going to draw with them? Water back into the blood. And it happens because the water on the thin descending is following the sodium and chloride that's being pumped back into the tissue fluid and the blood via the thick ascending part of the loop. Your distal convoluted tubule has a little bit of reabsorption happening. Mostly, of course, there is a lot of selective secretion at the distal convoluted tubule. But there are some important components to the distal convoluted tubule that we need to talk about, and that's these guys right here, the googly eyes, the way I think of them. They were my Facebook page for a while. So we have these guys called principal cells, and we have these intercalated cells, and the principal cells along the distal convoluted tubule and the collecting duct are going to be responding to aldosterone and ADH and PTH. So let's talk about it. Principal cells receive this hormone aldosterone, and what does aldosterone do? It says to reabsorb sodium. Well, getting rid of potassium, because our potassium levels are high inside our cells, our sodium is high outside our cells, and we must maintain that difference for all of our electrically active cells. And what are electrically active cells?
Electron transport chain? Electron transport chain?
Yes, but even more so, I'm thinking whole cells.
Like neurons.
Neurons and
Muscle.
muscles, muscle cells, all three kinds, smooth, cardiac, and skeletal. And if our potassium gets outside and is too high, we don't have that, right? It's serious when it comes to cardiac muscle, if your potassium levels get out of whack. And so, helpful getting rid of that extra potassium. Where did the extra potassium come from? Remember, it was reabsorbed along with sodium and chloride in the ascending limb. ADH causes alkoporons to be created in the distal convoluted tubule. So we pull water from the tubular fluid back into the tissue fluid and then back into the blood. And how does PTH work? So it has something to do with calcium. Remember PTH, parathyroid hormone? All about stimulation of parathyroid hormone is low calcium levels. So if we have low calcium levels, our PTH, our parathyroid hormone, is going to make sure that we pull as much calcium that was secreted, filtered out, get it back into the blood. Yeah, very important. So the principal cells are really cool. And then we have these intercalated cells, and the intercalated cells come in more than two types, but that doesn't matter. You're just going to know about intercalated type A and intercalated type B. And if we have a low pH, then those type A cells are going to get rid of, secrete hydrogen, and retain bicarbonate. And you know what? I've tried to explain this in the past. It's really complicated. It's such a contorted, tortuous mechanism by which this all occurs, so don't worry about it. Just know that our intercalated cells are working the pH aspect for us, and the type A cells are going to get rid of extra hydrogen ions but retain bicarbonate. Where does hydrogen and bicarbonate come from? Let's think about that. Carbon dioxide and
Water.
water. And this, by the way, goes both ways. CO2 plus water forms carbonic acid. dissociated bicarbonic anhydrase into bicarbonate and hydrogen ions. The bicarbonate and hydrogen ions are put together by carbonic acid and put into carbonic anhydrase and turned into carbonic acid, which then dissociates into CO2 and water. It goes both ways. That is such a flexible and a formula that has shown up, am I right? Time and time again. Yes, and when we start talking about pH mechanisms of maintaining our pH, you'll see it again. So we have the type B cells. They are going to respond to high levels of bicarbonate, and they're going to get rid of that extra bicarbonate and retain hydrogen ions to manage our pH, keep it within a narrow range. So aren't kidneys amazing? Look what they do for us. These little organs, silently just buzzing along, filtering, absorbing, dumping. Regulating. Yes. So that's the end of part 2. We just have a few minutes. We're going to take advantage of those few minutes.